Thank you. And it's a real pleasure to be here. I appreciate the invitation. And I'm sorry I couldn't be here yesterday because of that cholinergic mechanism. So I was in another meeting. So I'd like to talk about sex-specific small RNA regulators of acetylcholine signaling. And I'd like to try to bridge some of the data that we've heard this morning, and you probably heard yesterday, in relating the neurotransmission pathway to, on the one hand, to the small RNAs that are affected, including both microRNAs and transfer RNA fragments, which I understand were covered already yesterday, and try to show you how these pathways link together, and with a focus on men-to-women differences. So, a small task. Okay. And I'd like to start by telling you how I got to make all of these connections. So, this was my late PhD supervisor, Uri Litauer, who gave me a PhD project. I was supposed to synthesize a small fragment, about 1/4 of tyrosyl tRNA. And that was because at that time, tRNA was the only RNA you could sequence, because that was before the Sanger sequencing. And he published a paper on bacteriophage-induced transfer RNA in E. coli. And today, when I look at it from a distance, you see that bacteria react to infection by breaking down tRNA, sending it out. So this is quite reminiscent of the CRISPR statement, right? But nobody, of course, thought about that at that time. So what I was supposed to do required a purified enzyme to make the synthesis happen. And I purified polynucleotide phosphorylase. I can tell you proudly that Sigma still sells it under my protocol, so it holds. And that enzyme can work both ways. It can synthesize RNA, but it can also degrade RNA depending on the salt concentration, which is something I used based on an EMBO course that I went to in Brussels, where Sir John Gurdon was teaching us how to inject RNA into oocytes. The only RNA you could think would be clean was globin RNA, because erythrocytes make only globin. So what I did was to use my purified enzyme to chew into the RNA, remove the 3' end, and that led to the demonstration that poly-A tail stabilizes messenger RNA, which became my PhD rather than the left bottom quarter of tyrosyl tRNA. Okay, then I got trained in the Rockefeller University by Jim Darnell. I learned to do molecular cloning. I came back to Israel and I looked for a focus within the nervous system, which I did regard, and I still do, as the last frontier. I mean, this definitely is the most difficult one, but maybe also the most worthy one to look into. And I picked the cholinergic pathway, again, because we knew much more about that pathway than any other neurotransmission. Perhaps this is still true. So the idea was not to look only at the chemicals and proteins involved, but also to find out the RNA controllers. And in that aspect, I need to cite Kawashima from Japan, who talked about the expression of all of the cholinergic genes in white blood cells, and of course Kevin Tracy, who told us that acetylcholine blocks inflammation in those blood cells. So that gives us the concept, and now let's move on to gender differences or sex and gender differences. So back in 2018, there were 2 papers published in Science by a huge consortium of American leading scientists that said, forget about schizophrenia, don't think about bipolar disorder, there is a spectrum. Just like autism, there would be a spectrum of being mentally unwell. And I read these papers, one on transcriptomics and one on genomics, with great interest. There was no differentiation between men and women. But we know that men develop schizophrenia earlier. We know that males, schizophrenic males, get addicted to smoking, meaning the cholinergic pathway is highly relevant, but not women. We know women suffer much more drastic symptoms if they are bipolar affected. So what about that? So I went into the papers and I separated the data sets into males and females, and then the spectrum didn't look that nice. So that made me really upset because it meant that it wasn't a coincidence. They tried to present the data as nice as they could, and that required ignoring differences between men and women. So we went into those data sets to look into the cortical differences in short RNAs between men and women, and we remembered that women suffer more schizophrenia than men. I mean, those are all psychiatric experts. They knew all that. And it's a huge problem, like 3% of the human population. So this is the first chapter I'd like to talk to you about, data from 139 schizophrenic cortical tissues, 82 bipolar. And what we've done was to combine in-depth analysis with experimental validation of the pathways we looked at, thinking, are there differences between men and women? Can we find them by meta-analysis into the cholinergic pathway? Can we reduce the pathway to other processes? And hopefully with that, one day help to find medications that would be targeted not by developing drugs for half of humanity that are tested in the other half, but properly. Okay. So what we started with is a quadruple comparison between schizophrenia and bipolar and between men and women. And of course, we label men by blue and women by pink, remembering that 100 years ago it was totally the opposite. But okay. And what you can see is that in schizophrenic brains, we see very different pathways to be affected in the blue and the pink sections. Whereas in bipolar below, you see the same pathway is affected, but in opposite directions. And if we look at specific pathways, we see response to organophosphates, which is much talked about in the news because that's still the poisoning preferred by the Putin management. And that also meant that inflammation might be affected. So Sebastian Lobentanzer was a PhD student then, today is a postdoc in Heidelberg. And what we've done was to seek single-cell RNA sequencing of cholinergic neurons from human cortices or mouse cortices. There's much more data on the mouse. And we picked those because they express cholinacetyltransferase, so they are really cholinergic. And we found the genes, about 1/3 of the small RNAs are primate-specific, meaning that mouse, although it's very important, would not be enough to look into the processes. And we found no nerve growth factor. So you see the picture of Rita Levi-Montalcini here, because she found in the deep cholinergic nuclei that the cholinergic neurons depend on NGF, but the cortical family is very different, no NGF, and yes, association with inflammation. So Geula Hanin, she's now a postdoc, no, a young faculty member in Cambridge, UK. And she looked at the process, and what these neurons do is to work by the collection of neurokines like interleukin 6 and affecting the GP130 receptor to induce cholinergic messages. We found different microRNAs that Geula proved target acetylcholinesterase. So all microRNAs are equal, but some are more equal than others. And we could proceed to look into the microRNAs involved in general. We did that in cultured human neurons, neuronal cell lines of human origin. And that brings me to another point. We all know that HeLa cells came from a woman because the story of Helen Lane is known. But what about other cell types? I've been using cultured cells for decades, and I rarely think whether they come from a man or from a woman. And we decided to make a comparison. So grow one cell of this, one cell line of that, and find the microRNA differences. And what you see here is the blue ones are male-specific, the female ones are the pink ones. There are similar microRNAs, there are opposite microRNAs. There is a very different profile. So when you look at cells from now on, please think, where do they come from? Okay, so if we do that, what we see is that a woman-derived neuroblastoma cell produces mainly female-specific microRNAs, especially when it differentiates into cholinergic features. And a lot of the targets of those microRNAs we know from the literature as associated with schizophrenia or bipolar disorder. So here you are. You see the little pink dots go for female-specific ones, the little blue dots to male. And you see here that in schizophrenia, we have a lot of male-specific changes in the targets of these microRNAs. In bipolar, we have a lot of female-specific ones. And in the center, we have a mixture. So I definitely agree with the concept of one line, but one line for men and one line for women would be much more correct. After we published that paper, we were asked to submit a review on the male to female differences to trends in pharmaceutical sciences. And then I found that, for example, in our state, in the state of Israel, cholinesterase inhibitors have been used for decades to treat all Alzheimer's patients, like in many other countries in the Western world. But last year, suddenly, the dose for women was cut by half, because nobody knew before that women have much more severe side effects, because women tend to less participate in clinical trials than men. And then we have side effects that are distinct for men and women, such as calcium depletion from bones, which is very dangerous, or drug-induced psychosis, or a major depressive disorder treatment that might be very different between men and women. So this review is available. It was written by an MD PhD student in the lab, Alon Simchovich, who's now specializing in oncology. That's for microRNAs. What about transfer RNA fragments? So over the past decade, there is a revival of interest in these fragments that were thought before to be just a debris of the tRNA. Some of them are essential for sperm development, some would be essential for ribosomal biogenesis, and angiogenin is one of the inducers, and angiogenin levels, we know, elevate in patients after ischemic stroke, but nobody knew why. So angiogenin could reflect a change in these transfer RNAs. What do we know about them? There's the cloverleaf tRNA. It can be broken sideways or in the middle and create different type of fragments. And it may come from genes either in the nucleus or in the mitochondria, which is important. And there's a lot of genes for any of those. So think about a comparison. For like 10 years or so, I've been going around saying microRNAs are wonderful. They are so smart, they are small, they can be produced rapidly, they solve a lot of problems. Someone should have invented them. But then thinking about the production of microRNA, you need to transcribe it. That takes time. Now compare that to transfer RNA fragments. All you need is a hammer to break it down to pieces. It's always there. There are dozens of genes for each one of them. So we can look at them. Unfortunately, the nomenclature is horrible. The only clear thing is the number, which is the length of that fragment. But someone will get a better nomenclature soon. So we looked at those. We, in that case, is Katarzyna Winek, a postdoc in the lab, MD, PhD from the Charité. And we looked at them in blood cells from patients. And we focused on the cholinergic targeting ones, because transfer RNA fragments can work just like microRNAs. So the red dots show you the cholinergic targeted one, each of those targeting at least 5 genes from the cholinergic pathway. And what you see on the left-hand side are transfer RNA fragments, the levels of which increase dramatically within 2 days after an ischemic stroke in blood cells of patients, whereas microRNAs go down. So there is what we called a changing of the guards. MicroRNAs might be good enough to handle controlled processes under normal circumstances, but when you are in an emergency situation, you need to produce something more effective. And that's what the transfer RNA fragments do, definitely for the cholinergic system. So which transfer RNA fragments are involved? Many of them were actually thought to be microRNAs before people realized that they are coming from a tRNA gene. So here is one that can control stem cell development. And 2 of those seemingly microRNA-like transfer RNA fragments were induced in the blood of patients. And what Sebastian could show by analyzing their targets is that they target very different transcription factors. Namely, they would induce production of more cells of a certain type that you don't see otherwise. And the cells would be CD14 cells, and that raised the question in our mind, could these be new therapeutic targets that no one has thought about? And what we've done was to take mouse cells in culture, human cells in culture, prove that the transfer RNA fragments are induced in both, find one that targets a transcription factor, induce a mimic, and show that with that mimic, the target of that TRF goes down. So yes, indeed, we now have a novel family of potential therapeutic agents. Would that be relevant to other conditions as well, and to the brain, not only to blood cells? From now on, this is totally unpublished data. So we know that women are more susceptible to Alzheimer's disease. When they get sick, they deteriorate faster, both cognitively and physically. And what we've done was to compare short RNA sequencing files from women and men with Alzheimer's disease. And what you see in this slide is that the transfer RNA fragments, which is the top row, show huge differences that are particular to women, whereas the microRNAs don't show that. And there is a big struggle with microRNAs in the Alzheimer's brain for years now, and indeed it didn't go anywhere. So this is new and exciting, and we have a computer student who moved into biology, Dana Schulman, who's continuing this test in the lab. And where they come, where the TRFs that are modified come from, is both the nucleus and the mitochondria. And men do not show these changes. So TRFs in the blood may be good helpers to overcome the immune difficulties after stroke, but in the brain, they might cause damage in the Alzheimer's disease patients. We looked at the potential targets of those TRFs, and we find that many of them are indeed cholinergic for the changes in the nucleus accumbens, which is a cholinergic region. And not only that, we have the data, thanks to David Bennett and the Rush Center in Chicago, we have the data on the cognitive decline of these patients. And the changes in TRFs agree with or associate with the cognitive decline. Last but not least, and I was really interested to listen to the previous talks on the pre-birth stress. And we collaborate with Marta Antonelli from Argentina, who got a grant to run this study in Munich. And what they collected is umbilical blood cells from newborns, males and females, of women whose stress levels were tested psychologically before delivery. And the question is, what are the molecular processes involved? Are these microRNAs, transfer RNA fragments? Who is suffering? And look at that. So TRF profiles differentiate between stressed and controlled females, but not males. So there are differences in the newborns, but only females. I was totally surprised, but that's where we are. So differentially expressed TRFs. Where do they come from? They are all iTRFs, so one subsection, one nuclease is causing that. Again, no changes in the male newborns. And which tRNA do they come from? They all come from aspartate tRNA. So it's not a coincidental effect. So again, female, not male. And mainly mitochondria. We do know that females show much more mitochondrial stress in different situations, so this is true also for pre-birth changes in babies. And in summary, we would say that female newborns are particularly sensitive to prenatal stress, and that is reflected in the specific cord blood TRF changes. The TRFs present potential biomarkers. They could be therapeutic targets for addressing the damage caused by the stress. The babies are being followed these days for psychological changes, and we do find papers telling us about parallel things in the mouse. So I would say that in blood cells, TRFs may be SOS helpers, but in other situations like Alzheimer's disease, like prenatal stress, they might be causing damage, and that in any case, one must study them separately for men and women. I get a lot of papers to review, and I always say that, and then the authors say, but then we will lose our statistical significance. But now you have significance, but it's worth nothing. So Marie Curie told us that nothing in life is to be feared. She proved it. It is only to be understood. Now is the time to understand more so that we may fear less. I don't know, I get scared when I listen to the news, so I don't know to what extent we can reach that, but I'd like to thank everyone who did the study. I tried to mention the names along the way. The patients who donated samples, which is crucial for all of this, the supporting agencies that enabled it, my family members who suffer my passion for science for many years, and you for listening. Thank you very much.